Good morning to all my friends and my subscribers. Today we are going to study about a very well-known topic, which is a glycolysis. And this topic is mainly come in every life science exam. Even always come in CSIR net, set, and all other competitive exam. This is a very basic topic. Today we are going to see about the glycolysis. As we all know that glycolysis occur in every living cell, and it is a metabolic process, and it is made up of the Latin word glycose and lysis, mean the lysis of the glucose. The name itself indicate that there is in this process there is a lysis of glucose molecule. And why the lysis of this glucose molecule is there? To make ATP. And we all know that ATP is the energy currency of our cell, and our whole body run through this energy currency. So the first this pathway is called by the name of the scientist Embed and Merhoff, and these two scientists uh, discovered this pathway. So this pathway is also called as the EM pathway or Embed and Merhoff pathway. And the glycolysis process mainly occurs in the cytosol of the cell. We all know that the cytosol is the main part of the cell where many reactions are going through at a time, and one of these reaction is the glycolysis. So glycolysis occurs in presence of oxygen and also in the absence of oxygen. So both we can see the glycolysis in aerobic condition and anaerobic condition. So in the presence of oxygen, the glycolysis occurs and the end product is CO2 and H2O. And in the absence of oxygen, the end product is pyruvate and lactate. So these two things we have to remember, that the glycolysis end products. So in any competitive exam you have been asked that what is the end product in anaerobic and aerobic reactions in the glycolysis pathway. Let's see the next slide. And as I say earlier, that the glycolysis is the major pathway for ATP synthesis in tissue lacking mitochondria, erythrocyte, cornea lens, etc. So it is a major pathway of ATP synthesis. And we all know how the ATP is synthesized by the chemiosmosis mechanism. We all study that. But we have to see in this uh, at this time we have to see how the glucose molecule is converted to the pyruvate, and during this conversion how many molecules of ATP has been generated. And we all know that the glucose is the very basic source of carbohydrate, and this glucose source is very easy to break down. So this pathway is performed by every cell of our body, not only by the tissue lacking mitochondria, but every cell perform the glycolysis. And this glycolysis is very essential for our brain, which mainly depend on the glucose source as the energy. And the reactions of glycolysis. The glycolysis is divided into three distinct phases. First is the energy investment phase or the priming phase. Second is the splitting phase, and third is the energy generation phase. And I am going to teach you the glycolysis in three steps, and these three steps are very important to understand the pathway. And if you understand this basic pathway of glycolysis, then you never gonna uh, get problem in solving the questions related to the biochemistry. And we all know that the biochemistry plays a very crucial role in every competitive exam. So we have to prepare like this, and if our basics are clear regarding the biochemistry, we can excel in any exam. And you are thinking that how this glycolysis is so much important, or this EM pathway is so much important. But students, this is the very basic foundation of the biochemistry. And if you have summarized This pathway, no one can beat you in any competitive exam. So let's see the glycolysis process. So first we are going to see how the glucose enter into the cell. The glucose enter into the cell by the GLUT4 transporter, and every molecule when enter in our body, it requires the transporter, because we have to move. Even we require the transporters to go from one place to another. So this is the glucose molecule require the transporter, and the GLUT4 is the transporter for the glucose molecule, and this transporter transport the glucose from extracellular fluid to muscle cells and adipose sites, and this is the main site by the entry of glucose. And when the glucose enter in these cells, muscle cells and adipose site, but the question remains that why muscle cells and adipose sites? Students, muscle cells require high amount of ATP. Our whole body is made up of muscles, and if the muscle get the ATP, we move. So it is a very basic concept that the muscle require the glucose, and we know that if we don't have the glucose, we feel very low. We feel no energy in our body. So glucose is the main source of energy, and the energy, not the energy source, it is the main source for ATP synthesis, which is the energy currency of our body. And all this process of entry of glucose is take under the influence of insulin. We all know that the insulin is a very important hormone which which may function as a glucose regulator also. So a condition in which, like diabetes mellitus, insulin deficiency hinders the entry of glucose into the peripheral cell. And students, the diabetes mellitus leads to the insulin deficiency, and when the insulin deficiency is there, there is very low demand of glucose and no entry of glucose in the cell. So the person suffering from diabetes mellitus feel very low energy. Low energy means the person feel weakened. The weakness in the body is there in the person. And the GLUT2 is the transporter in the liver cell. So first GLUT4 transport the glucose into the muscle and the adipocyte cell, and second is the GLUT2 is the transporter in the liver cell. Remember these two terms, and these are the very important, GLUT4 and GLUT2. These two transporter may be asked in any competitive exam, like that the question will be framed, name any which in the following, which of the following is the transporter of the glucose, and you get the options like GLUT1, GLUT3, but you have remember the GLUT4 and GLUT2. These two you have to remember. Understand? The next we see the main part of our interest. The main part of our interest is the pathway. And students, we see how this pathway works. So first there is the entry of glucose in the cell. And when there is entry of glucose in the cell, the glucose is converted into glucose 6 phosphate by the enzyme hexokinase or glucokinase. And this enzyme is very important, students, because it is also the regulatory enzyme of the glycolysis. So remember the first enzyme, hexokinase, also called as glucokinase, which is the regulatory enzyme. Then glucose 6 phosphate is converted to fructose 6 phosphate by the enzyme phosphohexokinase isomerase. Then the fructose 6 phosphate is converted to fructose 1,6 biphosphate by the enzyme phosphofructokinase. And students, you have to remember the end product and the enzyme, because enzyme and the product both are linked together. If you memorize the product, you will easily memorize the enzyme involved in this. So, and we are seeing the first phase of the glycolysis. It is the first phase of the glycolysis. And fructose 1,6 biphosphate is converted to two molecules, dihydroxyacetophosphate and glyceraldehyde 3 phosphate. These two molecules are very important for movement into the second phase of the glycolysis. So before moving to the second phase, we require two molecules. We require two molecules of glyceraldehyde 3 phosphate. So we get only one molecule. So how will we get the two molecule? The dihydroxyacetophosphate, you remember, it is the product by conversion of fructose 1,6 bisphosphate. So dihydroxyacetophosphate is converted to glyceraldehyde 3 phosphate by enzyme phosphohexokinase isomerase. And this, then in the second phase, the glyceraldehyde 3 phosphate two molecules enters, and these two molecules of glyceraldehyde 3 phosphate get converted to 1,3 bisphosphoglycerate, and the enzyme involved in this is glyceraldehyde 3 phosphate dehydrogenase. Remember, you have to remember the enzyme also, and also the cofactors like NAD is converted into NADH. Then 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate, and the enzyme involved is phosphoglycerate kinase. And students, see here, the ADP is converted to ATP. This is a phosphorylation of ATP. So which type of phosphorylation is there? It is the substrate level phosphorylation. Remember, there are two reactions in the glycolysis which having the substrate level phosphorylation. This is the first reaction of substrate level phosphorylation, and second we will see in the third phase. So the 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme mutases. Next, the 2-phosphoglycerate is converted into phosphoenol pyruvate. And the phosphoenol pyruvate, how it get converted? So focus here, enolases enzyme is involved. And also remember the cofactors Mg plus 2 is also involved there. The phosphoenol pyruvate is then converted to the last product, or you can call the second last product in the anaerobic reaction, but last product in the aerobic reaction, which is AT phosphopyruvate. And here is the second reaction of substrate level phosphorylation, and the phosphoenol pyruvate is converted into pyruvate by the enzyme pyruvate kinase. Remember the last enzyme, pyruvate kinase. And in the anaerobic process, as I say, the end product is lactate. So after the formation of pyruvate, there is the formation of lactate by the enzyme lactate dehydrogenase. So remember student, all you have to remember all enzyme and all the cofactors. If you don't remember the cofactor, you don't have to worry, but remember the enzymes and remember the important reactions. And these important reaction will play crucial roles, like these two substrate level phosphorylation reaction. First is the glyceraldehyde first is the 1,3-bisphosphoglycerate to 3-phosphoglycerate. It is the first substrate phosphorylation reaction. And the second is the phosphoenol pyruvate to pyruvate. So remember students, these are the two important substrate level phosphorylation reaction, and the question related to this been asked in the exam. So at last we are going to see the regulation of glycolysis, and there are the three regulatory enzyme in the glycolysis process. First is the hexokinase and glucokinase, or both are the same. As I say earlier in the starting of the pathway, that hexokinase plays a regulatory function in the glycolysis. The second enzyme is the phosphofructokinase, and students, you remember where this enzyme comes. And the last is the pyruvate kinase, and we see the pyruvate kinase in the second last reaction, or the last reaction in the aerobic process. So remember students, and these three enzymes play a crucial role, and these are the catalyzing the irreversible reaction regulated in the glycolysis. So at last we are going to see, we see all over the pathway, all pathway we see, from glucose to pyruvate and lactate. But you have been saying that how many ATPs are being generated there. So my students, one molecule of glucose enter, then we get in the glycolysis we get two ATP molecules. How many molecules we get? Two ATP molecules we get, pyruvate we get, two NADH we get. And this is the energy currency we get. From one glucose molecule we get two ATP molecules. So that's all with my the glycolysis video. If you have any doubt or difficulty, comment it in the comment box. I will try to solve your difficulties, your doubts, and I will continue this doubt in the other video. And at the last of the other video, I will review your doubt and clear your doubt. So thank you for watching my video, and if you like, then forward this video, share this video, and like and subscribe to my channel. Thank you.